Hi everyone, I want to walk you through the steps we used in the Transformation of Bacteria lab as a review exercise. This is one of the concepts you really want to know well for the test that's coming up. So let's start with this. We're talking about a jellyfish, and this is the jellyfish that produces the green fluorescent protein, so it glows green. And what we did in this lab is we took that gene, we cut it out of the jellyfish genome in the cell of a jellyfish, and we inserted that gene in some way into a bacterial cell, and so these are then E. coli bacteria, glowing green. So transformation is all about taking a foreign piece of DNA and having a cell take that up and begin to express the genes that are on that foreign piece of DNA. Okay? All right, the method. This really kind of breaks down into two steps. One, you have to make what's called a recombinant plasmid that has your gene of interest on it. And then the second step we'll talk about on the next slide involves getting a cell to take up that plasmid. Okay, so the first thing you're going to do is, we have jellyfish genes. Okay, there's lots of genes in a jellyfish cell. Okay, so this is our jellyfish cell, and we actually need to we need to find the one that is associated with the green fluorescent protein, and we need to cut it out. Okay, so we're going to do that using a restriction enzyme. A restriction enzyme, we're going to say, looks like this. I think I just messed this up. Restriction enzyme looks like this. It cuts DNA at a specific location. And then we need a plasmid DNA. So plasmids are only found in bacteria, and they're circular pieces of DNA. We can cut that open. Okay, now convert that to a linear piece of DNA. And then we're going to combine these two things. We're going to combine our linear piece of DNA and our other linear piece of DNA that contains a green fluorescent protein gene into what's called a recombinant plasmid. Okay, we do that using restriction enzymes to cut, and we use DNA ligase to glue them together. Our final plasmid that we used in this case has a gene for green fluorescent protein. I'm just going to abbreviate that, green fluorescent protein. It also has a second gene on here for ampicillin resistance. Ampicillin is a bacterial killer. It's an antibiotic, and we'll see why we need that. But remember, the plasmids we used in this contain both of those. The green fluorescent gene, or green fluorescent protein, is kind of our gene of interest. That's what we're interested in doing. It could be a human insulin gene. It could be a human growth hormone gene. There's lots of other possibilities of things we might want the bacteria to make for us. The ampicillin resistance gene is just there to help us select the bacteria that have become transformed. Okay, so we're going to move on to the next slide here. In this one, I just want to indicate here real quickly what we're trying to do. So here's our bacterial cell. Right, it's got this bacterial chromosome here. Okay, we want our plasmid. Let's say this is our plasmid. And it's got the green fluorescent protein gene on it. We want that to go into this cell. So what we do is we treat the cells, the bacterial cells, with a hot and cold treatment in calcium chloride, and hopefully we get some cell to take up this plasmid. Okay, and so it'll look like this. It has in it now the plasmid for the green fluorescent protein. Oops, it should be green, right? Looks like this. Okay, also has this other gene on it for the ampicillin resistance. All right, so there is our cell that's taken up the the green fluorescent protein plasmid, and we would say that this cell now is transformed. Okay, so what we did in our lab was we added bacteria and we added plasmid DNA. We mixed those together. And we expected some bacteria to become transformed. And it's actually a pretty rare event. That's why I only show just a few of these plasmids as being, a few of these bacteria as being transformed. Most of them are untransformed. So you can see here's the untransformed. We use this color, so we have a whole bunch of these that are untransformed guys, and just a minority that are transformed. The figure we use is about 1 in 10,000 cells, bacterial cells, become transformed. So it's a very low percentage. Now, if it's only 1 in 10,000 that become transformed and we want those transformed cells, we need a way of finding them. And so that is where the ampicillin resistance gene comes in. So we have our plates. These are agar plates. Then some of them had ampicillin on them and others didn't. No ampicillin. Okay, so what we did was we took our solution here that contains these, so this is a solution. We plated some of this. We put some on that plate and we put some on this plate. Okay, if there was no ampicillin in the medium, then all bacteria, transformed and untransformed, could grow here. And so we see what they generally call a lawn of growth. So it's just bacteria everywhere. Okay, because there's no ampicillin, nothing to kill them. Now, on the other plate, though, it's a little different. There's ampicillin here, and so that's going to kill any bacteria that don't have the ampicillin resistance gene, which is 9,999 out of 10,000. Almost all of them are not going to have the ampicillin resistance gene. And so rather than seeing this lawn of growth like we see here, we're only going to see a few isolated colonies. And what these are, they represent individual cells that had become transformed and then landed here and started to divide from 1 cell to 2 to 4 to 8 and on and on and on until we get a visible clump of cells there after 24 hours that look like this. Okay, so that's a colony of cells, and they all descend from that one original cell that was actually transformed. Okay, so that's what we're using the ampicillin for, is to get rid of all of these guys. They all die off, and all we get are the ones that we're interested in, the transformed cells. And then we can continue to produce more and more of those cells and that gene, the gene of interest, and whatever that gene product is, the protein, for human use if it's a medication we're trying to produce, or if it's some other protein we want. This bacteria now will produce that, and we can collect it and purify it, and there's a lot of possible uses for that. But this is the basics of our transformation lab. I hope that makes sense. Go back and watch it again if it didn't, but you want to understand this in pretty good detail for the test.